Hi, I'm Fei Chen, and I run a group here at the Broad Institute. And our group is quite interested in developing spatial genomic tools that allow us to measure the molecular profiles of cells directly in the context of tissues. And as part of this work, funded by the NHGRI, to develop approaches to generate high resolution spatial transcriptomic data, wherein we can use high resolution capture arrays to profile the gene expression either by directly capturing the RNA and DNA molecules or in recently published work using something called slide tags, where we use these arrays to barcode cells, and then we know where those cells came from in the tissue. And this allows us to understand how cells communicate, how their gene expression changes as a function of their environment, and how they interact in both health and disease. So today, we're excited to tell you about our work, really led by Chenlei and funded by this opportunity fund by the Genome TDCC that sets out to do kind of a really cool thing for spatial transcriptomics. And the motivation and idea behind the project is twofold. One is to really democratize access to spatial transcriptomic technologies. And two, it's to demonstrate this new conceptual approach for generating spatial data from sequencing data only. And so with that, I think I'll hand it over to Chenlei, who will tell you about how this technology works and what we can learn from it.
I'm Chenlei, and I'm a graduate student from Harvard University working in Fei Chen's lab. So our lab has previously developed slide-seq and slide tags, and they are spatial transcriptomic methods that utilizes spatially barcoded array. These arrays are made of DNA-coded beads here, where each bead has a unique spatial barcode. And to correlate the DNA barcodes to the beads to their physical locations, we set up an imaging system to perform the in situ sequencing with the arrays. Then the big barcode can be read out base by base in situ. The imaging system is actually difficult to set up, and it also limits the size and the throughput we can make with the spatial transcriptomics method. So this limitation actually motivates us to develop an imaging-free spatial transcriptomics method. It is kind of counterintuitive that we try to figure out where things are without looking at them, but there are many interesting examples that inspire us. For example, in biology, Hi-C measures the frequency at which two DNA fragments physically associate, and this reflects their physical location in 3D space. And also, if we know the distance between the US cities, we can actually reconstruct the US map. And here is another interesting example, shows that genes actually mimics geography. Here, each data point is an individual's genotype. And by projecting them to a two principal component embedding, people find that actually the geographical map of the Europe arises. So all these examples suggested that spatial locations can actually be reconstructed from spatially related variables. This motivates us to develop our imaging-free spatial transcriptomics method. And here we use diffusion as a local spatial variable to infer the physical locations. On the same structure of original slide-seq or slide tag bead array, we mix it with diffusible beads. These diffusible beads have a photocleavable linker that allow them to be released and start to diffuse upon UV exposure. Also, they have this reverse complementary poly-A tail that allow them to bind to the capture beads. By sequencing the conjugated library, we can get a diffusion matrix which tell us the frequency of the interaction between two different barcodes. With the diffusion matrix, how to reconstruct the 2D physical location is actually a math problem. When we look at the high-dimensional diffusion matrix, we find it quite similar to single-cell data, which is a cell by gene matrix. In single-cell analysis, we often use dimensionality reduction to visualize the single-cell data in 2D embedding. In this process, cells with similar gene expression profile are still in proximity in the 2D embedding, and they can be clustered together as the same cell type. Same here, we want to capture beads that capture similar set of diffusible beads to be physically close to each other. Also, the high-dimensional diffusion matrix is actually generated from the 2D physical space and has an intrinsic 2D manifold. So we want to use dimensionality reduction to learn how the low-dimensional manifold. We started with simulation. We sample locations from image as capture beads and diffusible beads, and then simulate the diffusion process in silico. With simulated diffusion matrix, we apply UMAP to project to 2D space with different parameters and with the set of parameters that return uniform and circular results. We find the locations of the beads are successfully reconstructed and the image are well reconstructed. Then we apply this reconstruction method with slide-seq, and we find that UMAP with the experimental diffusion matrix can reconstruct the physical location with high fidelity, as the reconstruction result is identical to the ground truth. We also quantify the error at different measurement lengths, which show that the error is around 10 micron for local distance and below 3% for longer distances. We can also combine it with slide text to get the single cell resolution spatial transcriptomics data. We made very little change to the protocol. The locations of the nucleus are well reconstructed with very low error. Without the limitation from imaging, now the reconstruction allows us to scale up to large-scale spatial transcriptomics, and we applied it to a P1 mouse section. Here, the video shows the iterations in UMAP that is reconstructing the spatial locations. And without ground truth, we find it highly similar to an H&E staining of an adjacent slice. And we can also look at the details of the spatial distribution of specific cell types and specific gene expression. The fine structure of the tissue are all also well reconstructed. So here we presented this imaging-free spatial transcriptomics method where instead of imaging, now we just need molecular biology experiments to do spatial transcriptomics. Also, it's easier to scale up by just doing more molecular biology experiments. Also, we are quite interested in this idea of using UMAP, a dimensionality reduction method, to reconstruct the spatial location with the high dimensional diffusion matrix, showing that UMAP can learn the low dimensional embedding from the high dimensional data with quite high fidelity and making this tool practical for use.
We've open sourced these protocols as well as the computational approaches. And so anyone really can take this molecular biology reaction and generate spatial transcriptomic arrays. Because it's just a molecular biology reaction, we're no longer limited by the constraints of imaging, and sequencing costs are rapidly decreasing. So this allows us to have really arbitrary array sizes. And this becomes quite important as we scale to performing spatial transcriptomics on human organs, which are on the centimeter scale. My collaborator and I, Eben McCosco, are building arrays for entire human brains that are 7 centimeters in diameter. And if you think about it, that's 400 times the area of the first slide-seq arrays that we made back in 2019 and in a single experiment. And the other thing is that this sort of production is quite scalable. And so it's going to really increase the scale of performing spatial transcriptomic assays in health and disease.